My name is Dallas Abraham, and I'm a research associate at IMCS. I received my bachelor's degree in chemistry in 2018 from Union University, and then my master's degree in analytical chemistry in 2021 from the University of South Carolina, studying under Dr. Susan Richardson and analyzing environmental contaminants in water sources. I came to IMCS in 2021 as an application scientist before transferring to R&D to work on novel chromatography workflows on automated liquid handlers. One new product development is our SizeX product line, expanding into a new platform called Dynamic Devices. We leverage the unique feature of the Lynx, where it has a 5mm head and a 24 array. This pipetting mandrel can accommodate a larger pipette. A larger pipette obviously is conducive for larger sample volumes. These 1mm tips are typical and have been modified to desalt or buffer exchange. A large molecule can pass through the column, and small molecules like salts, dyes, and reducing agents will separate from the larger molecules. This is the general premise of size exclusion chromatography. With this larger tip, you can pack more resin, which would enable our customers to use a larger sample volume. So I'll demonstrate the SizeX1000 on Dynamic Devices Lynx with the 5mm standard technology mandrel to desalt 1mm of sample. A run to desalt 24 1mm samples in 10 minutes. It is by far the fastest desalting process, and extremely useful when you're under a time crunch. Examples of useful cases are affinity exchange followed by desalting to bypass acid neutralization in protein A eluates. Antibodies are typically eluted in acidic buffer from protein A resins. Instead of adding a base to neutralize the solution, you can transfer the eluate to SizeX and transfer the sample to a formulation of choice. Another one is imidazole eluates. When using IMAC columns, getting rid of imidazole in the eluate is also a time-consuming process. But here, for mid-throughput, having this automated would be beneficial. You can think of this SizeX as a quick way to run your buffer exchanges or desalt. IMEX has tested a variety of different molecules on SizeX for Dynamic Devices. You can see how the same method and product has been used to desalt viral particles, plasmid DNA, and polyclonal human antibody, as well as GFP. The recoveries across the board are consistent. Whether targeting larger biomolecules like antibodies or smaller protein like GFP, you will get some sample loss at the lower concentrations, which is typical for all desalting operations using size exclusion chromatography. So, if there are any other questions about the SizeX product series on Dynamic Devices, please reach out to our technical sales team or directly on our website at IMCStips.com. You can also send us an email directly at inquiries@IMCStips.com. We can also be reached at our toll-free number 188-560-2073.